Over the past 5 or 10 years, the field of antibody research has come to understand a couple of important things. One is that some HIV-infected individuals make remarkably potent antibodies against the virus, and researchers have been able to isolate those antibodies. So we have them in hand as potential clinical products now, and they can potentially be used either for prevention of infection or treatment of infection. So what we talked about with regard to treatment are that there are several areas where one might consider using antibodies. First of all, the recognition that antiretroviral drugs remain the cornerstone of treatment. So the question is, where could antibodies potentially play a role? And there were several that we talked about that I mentioned in my talk. One was that antibodies may play a role if given very early in infection with antiretroviral drugs to bring the virus under control very quickly and therefore have some impact on the latent reservoir. The other area would be to help maintain viral suppression. So once individuals are well suppressed on oral antiretroviral drugs, the question is how long can they be maintained, and is it possible to either withdraw antiretrovirals or move to a very limited regimen? And here there may be a role for antibody to help maintain viral suppression if antibodies can be made to have a long circulating half-life. And lastly, and potentially most importantly, would antibodies have some effect that antiretroviral drugs don't have? And that in one area that may be possible is the ability to kill HIV-infected cells. So antibodies have a well-known effector function that allows them to bring in close proximity certain types of immune cells that can kill virally infected cells. So if antibodies can mediate that kind of cell killing, they have a potential, if given with other types of treatment to, for example, induce expression of virus from cells, they may be able to kill cells and affect the viral reservoir.